off with Mitra Husman to give an introduction to the seminar. Oh, Mitra, you're muted.
Oh, thank you. Thanks, Ross. Good morning, everyone, and welcome. On behalf of all the organizers, I wanted to thank the wonderful panelists, the moderator, and of course, all of you for being here today. We look forward to a very productive and informative discussion. My name is Mitra Husman, and I'm sitting in for Sean Patel, actually, who unfortunately tested positive for COVID earlier this week, and he thought he would spare all of you from his stuffy voice, but little did he know that my normal voice isn't actually all that much better. But if you would just be patient with me for a couple of minutes, I wanted to briefly introduce CIRM and talk a little bit about our mission and how it fits into context with this particular webinar. So for those of you who may not be as familiar with us, our mission is accelerating world-class science to deliver transformative regenerative medicine treatments in an equitable manner to a diverse California and world. Next slide, please. And CIRM was conceived in 2004 when California voters passed Proposition 71, a bond initiative to provide $3 billion of funding towards the development of regenerative medicine therapies. Since then, CIRM has established a track record as a patient-centric funder, partner, accelerator, and de-risker for basic translational and clinical research infrastructure and education programs. Then, not too long ago in 2020, thanks again to the continued foresight of California voters, Proposition 14 passed, which reauthorized CIRM, providing $5.5 billion of funding towards the continued development of regenerative medicine therapies. Next slide, please. Shortly after the passage of Prop 14, we released our 5-year strategic plan with 3 main themes, which you can see here: advance world-class science, deliver real-world solutions, and provide opportunity for all. Under each theme, we have set very ambitious goals, the details of which you can read about further in our strategic plan. But what I wanted to highlight here, which is particularly important in the context of this seminar, is that across all these 3 themes, there is a systemic element with an objective of promoting knowledge sharing and collaboration across many stakeholders. This is from academic researchers all the way to manufacturing networks and industry partners and beyond. We're very excited about launching this strategic plan and believe that today's webinar is directly aligned with that objective of knowledge sharing and promoting these types of collaborations. So with that, I'd like to introduce you to my colleague, Ross Akamara, who, along with Sham, has organized today's webinar, and he will provide a brief introduction as well before we get into the wonderful discussion with our panelists. Ross, go ahead.
Thanks, Mitra. My name is Ross Okamura. I'm a senior science officer in the Therapeutics Development Group at CIRM, and CIRM has organized this webinar to discuss the development of pluripotent stem cell-based therapeutics. Since its establishment in 2004, CIRM has always been associated with the development of stem cell-based regenerative medicines, and that's easily shown to be true when you look at the funding we provided. Roughly 60% of our research and development awards, which is about over $1 billion worth of funding, has been to research utilizing pluripotent stem cells. And by that, I mean both human embryonic stem cells and induced pluripotent stem cells. Our funding pillars are indicated on this slide, as well as the definitions we apply to different stages of development. In our discovery stage, we ask our investigators to take what their knowledge of regenerative medicine and develop a single product candidate. At the translation stage, we fund what's known as the valley of death between bench and bedside. We fund activities such as GMP-compatible process development and scale-up, pilot toxin safety studies, the drafting of a clinical protocol, everything you need to make a package for your first formal interaction with FDA, a pre-IND meeting. After you get your pre-IND feedback and want to do your pivotal IND enabling studies, that's what our CLIN1 mechanism supports. And then after you have an IND, we support the clinical trial itself with our CLIN2 program. We also have listed here the amount of funding we put to each of these different areas. A lot of the funding has gone toward the discovery stage with 447 awards and $719 million of funding. And we have also 20 awards at the translational stage that we made and 14 at the clinical stage. And this is typical for a pipeline. What you'd see is that you have a lot of input, many candidates going into the process, and then some of the successful ones are the ones that you advance at the different stages. Additionally, what you see from this is that as you move through the pipeline, we've increased the funding for each stage, recognizing the additional costs that go into translation and clinical stage programs. For today's talk, we're going to focus mainly on the transition between discovery and translation, the development of a single product candidate. And the program we use to achieve this is shown on the next slide. And that's the DIS2 Quest Awards. And for this program here, the funding that we provide was previously $900,000 in direct costs for developing a therapeutic, but we're upgrading it to the next round to $1.5 million in direct costs. The duration of the award was previously 2 years. We're now allowing to propose up to 3 years worth of studies. And the activities we support during this mechanism are listed here. They're taken directly from our program announcement. It's easy to find the definitions there on our website. But just briefly, it's developing assays to identify and characterize the candidate, optimization studies of the candidate, characterization of it, proof of concept studies with the candidate. And we also ask the investigators to develop a target product profile at the end of the awards. We want them to draft one out. And this is the beginning of a product concept document that will grow along with the candidate as you take it through the discovery pipeline. And the end, what you end up with is a great tool to discuss the context of your therapeutic with FDA. We also fund what another appropriate cost in this stage is preparation for and conduct of stage appropriate regulatory meeting. And for here, we're talking about what used to be called a pre-IND meeting, but is now known as an interact meeting with FDA. And so that describes what the program supports. On the next slide, I just want to briefly say what the outcome is. And you don't need to read all of this, just the bottom bullet point that's bolded. For candidates composed of or manufactured from stem cells, reproducible disease-modifying activity is demonstrated with the candidate in a preclinical model relevant to the target indication. And for the purposes of the talk here, we're specifically talking about the phrase with the candidate. And for allogeneic pluripotent stem cell derived therapies, that's important because probably what the researcher has done up to that point has been using a research grade line, and we're asking them to show this proof of concept with a line that can be taken to the clinic. And on the next slide, we list in our program announcement exactly what we're looking for. So we're looking for mainly two things in the pluripotent stem cell line you choose. We want to make sure that you have informed consent to the line for clinical development and commercial sale. And additionally, we want to make sure that the donor you derived it from meets donor eligibility criteria as listed in Code of Regulations, Title 21, Part 1271, Subpart C, which is a great read. If you're developing a tissue therapeutic, you should understand what FDA considers to be a risk. This guidance, this regulation, along with the guidance associated with, you could find one from FDA, that indicates what that you have to do, what you need to screen for. The guidance suggests how you should screen for it. So there's a certain window you have to meet. It needs to be screened within, I think it's 14 days after donor donation. And also for diseases you can't test for, they also list populations that are more at risk for certain diseases. And you might not want to choose those populations for deriving your cell lines from. So I've described what an eligible line would be, and what we need to consider is where you get these lines from. And on this next slide, I've just listed some sources that CIRM grantees have used in the past. So we've seen proposals for using human embryonic stem cell lines from Y-cell, the H lines there, the ESI lines available from Ajax Therapeutics currently. And we've also seen proposals for the Hadassah lines from Israel. Additionally, we've had investigators use the NINDS line from Sampled, and also the lines from Fujifilm Cellular Dynamics have been proposed in our projects. And the next list that was just up there, we also are aware that some organizations, I've just listed a couple of California organizations here, have generated their own donor eligible lines for use by their investigators at their GMP facilities. And I'm sure there's more than this, but we're aware of at least the Cedars-Sinai facility, UCLA and UCSF have generated lines for their investigators. And the last option that we expect to see more of is that there are a number of biotechnology companies out there that are also offering donor eligible iPSC lines that they've generated in their GMP facilities. And I'm sure the list is longer than this, but again, these are ones that CIRM is aware of. And I just wanted to show you that there's a variety of sources that you can get pluripotent stem cell lines from. And the important thing about there being a variety of them is that they're called pluripotent stem cells, which means they have the potential to become any tissue in the body. However, anyone that's researched these lines and tested a number of them realizes that they don't all necessarily have the same potential. You can take a given differentiation protocol, but what you do if you test multiple lines is you'll see that that some may require a longer time to differentiate, some may require more growth factors or less growth factors or different growth factors. Some may require, or some may produce cells that aren't as efficacious as other ones. And so it's important to choose the right one. Recognizing this on the next slide, CIRM realizes that this is an additional cost for our investigators that are developing pluripotent stem cell-based therapies. So in the next round of DIS2, which is due in August this year, we are making available an additional supplement they could apply for, an additional supplement of $200,000 specifically for the purpose of screening and evaluation of multiple clinically compatible human pluripotent stem cell lines to select the final candidate for translation. And so we're trying to reduce the burden of taking this additional step that we think is important, is going through multiple lines and trying to figure out which is the right one for them. So on the next slide, I just want to introduce that as a way of why we've convened this webinar. Is we've invited speakers that have already gone through the process of developing a pluripotent stem cell-based therapeutic. And we're asking them to share their experience of how they went through this process. What sort of things should you look at? What type of differences can you see? What are good considerations for the stage of development that you're at? And through their talks, I hope to further this discussion. After the talks are over, we're going to have a question and answer period, and we have some prepared topics. But we're certainly looking forward to the audience providing some questions, and feel free to use the mechanism in Zoom for the question and answer panel and put questions in there, and we'll try to get them as many as possible to the panel to discuss. So as you listen to the talks and afterwards, if you have anything you want to ask, please go ahead and prompt us there, and we'll be looking for those questions. And with that, let's go ahead and finish with our slide deck, and I'm going to ask our first speaker, Dr. Geffen Millman, to give the first talk. As an introduction, Dr. Geffen Millman is currently an associate professor of medicine and biomedical engineering at Washington University School of Medicine. He received his PhD from MIT and completed his postdoctoral training in the laboratory of Dr. Douglas Melton at Harvard University. His current research is focused on synergizing both biomedical engineering and cell biology approaches to use stem cells for the study of and treatment of diabetes. He's developing new approaches for the differentiation of insulin-producing islets from stem cells and improving their utility for cell replacement therapy and in vitro disease models. His innovations have been licensed to several biotechnology and startup companies in the diabetes space and resulted in one ongoing clinical trial. His laboratory is supported by the NIH, NIDDK, and JDRF, and his scholarship has recently been recognized by awards from JDRF, American Institute of Chemical Engineering, and the Biomedical Engineering Society. And so I'll pass it off to you, Jeff.
Great, thank you for that introduction. It's great to be here to talk about a topic that I care a whole lot about, which is choosing the correct cell line and making sure that that can work for you. So I'm really hoping to share my experience here, both the good and the bad that we've had with this, and kind of the challenges that we see that can be discussed further during the discussion period. So I'm located in St. Louis at the WashU Medical Campus, and I have a team of people that come from a mixture of technical backgrounds, so with about half the lab being biomedical engineering in their background, and the other half being more biology in their background. And that's on purpose to try to get a diversity of ideas and experiences in order for us to really do the best that we can in terms of using stem cells for the study and treatment of diabetes. So to talk about that a little bit more, so diabetes is caused by the loss of healthy beta cells that are found in a tissue called the islets of Langerhans within the pancreas. So the beta cells are the cells that are responsible for producing and secreting insulin into the bloodstream that allows for proper control of blood glucose levels. And there's a lot of different mechanisms by which you can lose healthy beta cells, but the result is in the end is also the same, that you just don't have enough of these healthy cells in order to properly regulate in order for your body to normally properly regulate blood sugar levels. And so that's why the most common therapy used for patients with diabetes is injection with exogenous insulin. Essentially, the patient, either manually or using pumps or other technologies, is trying to do the job that the beta cells would normally do without you thinking about it. And it works. It's a life-saving therapy that a lot of people use, but it is very imperfect. It's just impossible for devices or individuals to manually do the job that the beta cells endogenously are able to do without you thinking about that. And that imperfection in being able to control blood glucose levels, it leads to a lot of complications that are shown on the slide here. And so kind of the goal of my research and other labs that are working in this space is to basically generate cells exogenously to replace the function loss by the endogenous cells. And especially as we think about this therapy kind of getting out of the research phase and being used more clinically, and realize how many people worldwide have diabetes, that really says that we need to have a large-scale sustainable source of replacement cells and tissues in order to begin to treat even a fraction of people with diabetes worldwide. And so my team, of course, uses stem cells as the approach here. We use both ES cells and iPS cells to be able to manufacture these cells, relying very heavily on the fact that these cells are pluripotent and can become beta cells. So on the screen here is basically a simple cartoon schematic of our current differentiation process. We've been publishing papers for quite a while now on initially figuring out how to make these cells and then iterating on the process to make the process a lot better. And this process takes about a month, so it's a fairly long manufacturing process here. And essentially what we're trying to do with this differentiation process is to mimic what happens during normal embryonic development, but doing that instead artificially in a laboratory inside of incubators. And so each of these stages are designed to give signals that the cells would normally see in the embryo to make the next stage of critical intermediate cell types and subsequent stages in order to make these cells. So it really is the combination of multiple factors usually in the correct sequential order that is the key in order for us to have a successful differentiation process. And so the very early work in this space has been used as the basis for an ongoing clinical trial that is being conducted by Vertex Pharmaceuticals. So getting back to the process here, we found that having really high quality cells during these intermediate cell types is very important. If you lose like a little bit of efficiency very early on in the differentiation process, that little loss of efficiency tends to amplify as you go through subsequent stages of differentiation. And so we have various quality control metrics that we can use to make sure that we are efficiently inducing all these intermediate cell types, such as FOXP2, SOX17 for end of stage 1 of the process, and PDX1 and NCAC61 that is done later on in the process. And so in the end here, if we're able to indeed have high quality differentiation through these intermediates, the end of the process will produce a final cell population for which the majority of the cells will be able to produce and secrete C-peptide or insulin. And the vast majority of them are endocrine of some nature, as defined as being a chromogranin A positive cell. And at least our way of doing this accomplishes this without using any sort of sorting or selection methodologies. So the end result of having high purity cells that are highly functional is that these cells are able to rapidly reverse pre-existing diabetes in a mouse model. So here I'm just showing, combining data from 2 different publications just to simplify things down here. This is stem cell derived islets made from a embryonic stem cell line in blue, or an iPS line here in purple. And you can see, again, there's a little bit of difference here by plus or minus a week, but overall, the reversal of diabetes in these mice occurs comparably between these two cell lines and on a rate that is similar to an equal number of cadaveric human islets that were transplanted alongside the transplantation here. And so we're able to get to, within a few weeks, have full reversal of hyperglycemia in these mice, and are able to maintain this this normal glycemic state for many months. I can only have about up to about half a year here, but we've in other studies been able to take this to the entire remaining lifespan of the mice, which is about a year. So this is both works fast. It is also long lasting for basically the maximum amount of time that we're able to do it using a mouse model. So to kind of start to wrap up things here, I want to talk about the challenges that we face with stem cell derived islets, particularly cell line variability. So this is a publication that we published last year showing success of our differentiation process with 9 or 10 different cell lines, depending on how you want to count it here. And the first thing I want to draw your attention to is that while we classified all of these as successful, there is still quite a wide range of variation here. So this is just a fractional yield of the beta cells. You can see that the fractional yield of the beta cells across cell lines can vary by up to 30%, which is a pretty huge margin. And we didn't show this in the study, but I can tell you that this variation is cell line intrinsic. So if you do like multiple batches of differentiation, then it would be consistent for that particular cell line. So there are indeed cell line intrinsic features that are causing the specific fractional yields that we're achieving with this differentiation process. Similarly, when we look at the function of these cells, so this is insulin secretion under low and high glucose levels, you can see that, again, these cells are able to secrete insulin. And again, there are some pathogenic lines here, but but we have several non-diabetic healthy lines here in the middle. So while they are able to respond to glucose and secrete insulin, again, there's a wide variation in terms of how they are able to do this, again, varying by approximately a factor of 3 here. And it's not just the beta cells that we see variation in. The other islet cell types that a process makes, such as alpha cells and delta cells, also have variation in their yields. And this is quantified down here. And there's a quirk about these cells where they can commonly be polyhormonal. So I've actually separated the populations here. You can see that for the glucagon expressing and the somatostatin expressing cells, I mean, they're present, but they can vary by quite a bit. And some cell lines like this AN 1.1 doesn't really produce a whole lot of somatostatin expressing cells. And so that adds an extra degree of complication here, because in general, we don't really understand the role of the non-beta cells in the context of this as a drug product for diabetes or replacement therapy. And so we don't understand it. And then we also have variation in it across cell lines there, which just basically compounds the stuff that we don't know about the cells that we have here. So one of the things that we've had to do in order to be successful across multiple different cell lines is that we commonly need to tweak the differentiation protocol to work. And by that, it's usually not really the factors that the key factors that we have in the process for actually like inducing beta cells versus other cell types towards the end of the differentiation process. The tweaks that we more often than not have to do are typically all in the very beginning of the manufacturing run. So basically how we handle the stem cells and how we seed down the stem cells. Seeding density is hugely important for this and varies wildly across cell lines. And then typically how we conduct the first stage of differentiation to make defendants Venoderm. Again, here I showed us having to resort to a kit in some cases, or adding in BFGF. We have found that to help out as well. So again, it seems that while we don't know biologically why really the underlying reason that cell line variation exists, this data would at least point to something very early stage during differentiation for which these cell line specific differences can cause problems in your ultimate differentiation process. So part of what we're trying to do right now in order to improve the manufacturability of these cells is that we've partnered with the Advanced Regenerative Manufacturing Institute in New Hampshire, where they have really great approaches and people for automated manufacturing of cell products, including differentiation. I'm just showing you some pictures of the process here, but we're basically trying to take out the human element here, having machines that can consistently perform the same actions to consistently differentiate cells across batch to batch in order to try to minimize variation that is caused by just humans being the people who are in the clean room doing the pipetting here. One kind of other aspect that I wanted to talk about real quick when it comes to cell line variation is that this cell line variation and the protocol that we are using to differentiate are linked. Basically, that variation can be increased or decreased depending on what differentiation protocol you're using. And what I have here is 3 different cell lines showing 2 different differentiation protocols. One is a protocol that we use cytoskeletal modulations to accomplish. The other one is a protocol that's completely done a suspension for which we don't do the same sort of modulation. So we have some cell lines like this embryonic stem cell line that didn't really care about which protocol we were using. So it was equally as good independent of the protocol. But we have other cell lines, and these are 2 IPS lines here, that just responded a lot better to the cytoskeletal modulation protocol and less well to the suspension protocol. So that's something else to remember as well, that variation may be better or worse depending on the exact protocol version that you are using. In our experience, we tend to have less variation and greater success with protocols that include cytoskeletal modulation. Part of this is that, at least if you're using a suspension-based protocol, we have encountered a decent number of cell lines at this point that just simply don't grow, as stem cells don't grow very well in a suspension propagation sort of system. This is just showing one of the cell lines that from BJ fibroblasts that we were never able to grow well in suspension, but we have gotten in other studies to work well when it comes to protocols that don't rely on that there. And so finally, just wanted to acknowledge, so even with how well the differentiation process can work in some cell lines, like other stem cell differentiation systems, our cells that we produce in vitro tend to be immature when you compare them to their in vivo natural counterparts here. So this is again an instant secretion assay. You can see that for both our 2014 and 2019 protocols that we are still secreting less insulin and have the cells being less glucose responsive compared against the real thing. This also translates over to marker expression. We can measure the cells and see a lack of markers that we typically associate with maturation of the cells. So in this case, MAPA and UCN3 are common beta cell maturation genes that are used that are just simply absent in stem cell derived beta cells, though we can increase the maturation of these cells after we transplant them. This is just showing on a very global level that the beta cells after transplantation for 6 months have a similar gene expression profile to primary beta cells that is very different than the in vitro, never been transplanted before sort of cells. So that's yet another thing that we as a field would want to improve in order to have a better drug product in the end. And that's really the takeaway here, is that I think that addressing these challenges will increase the or improve the manufacturability of these cells, decrease costs, and ultimately allow for people to get into the clinic faster or more efficiently than what we're doing currently. So with all that said, I want to thank my funding, particularly from JDRF and the NIH for my work there, my amazing collaborators, and my great team here at Washington University. And I'm looking forward to listening to the other talks and having a very productive discussion. So thank you.
Thanks, Jeff. I just want to let people know that we are seeing the audience questions. We're going to discuss them all together at the end of the sessions. We're just going to continue on with the talks right now. Our next speaker is Dr. Tomishima. He's a vice president of platform discovery at BlueRock Therapeutics, where he's been for almost 5 years. His role at BlueRock is to provide the strategic vision to advance BlueRock's cell and gene platform. Prior to the 5 years that he's been at BlueRock, Mark spent 16 years at Memorial Sloan Kettering, first as a postdoc with Llorente from 2002 to 2006, but later running his own lab, the SKI Stem Cell Research Facility, until 2017. His team led process and assay development and ultimately the GMP manufacture of MSK-DA01, a cell therapy derived from human pluripotent stem cells as part of a NISTEM funded consortium led by Llorente. DA01 is now the lead product in BlueRock Therapeutics pipeline and recently announced this week that they completed their enrollment for their phase 1 trial. So congratulations on that. That's a great step forward for you guys.
Thank you. Thanks for that introduction, Ross, and thanks for the opportunity to be here to talk about some of the work that we're doing at BlueRock. So what I'm going to do is start by talking a little bit about our platform, and I think a lot of those going through that journey will help to guide our discussion later, but I do intend to try to bring it back to this audience as well at the end. So at BlueRock, we call our platform the Cell Plus Gene platform. And on the left-hand side, you see the authentic cell. That's really the foundation of our platform. And we call these cells authentic cells because they're pluripotent stem cell derived products. And so therefore, they have the same biological characteristics of the cells that are degenerating in many disease states. So it's our hope that we can replace those cells in patients and restore function and hopefully reverse disease. Now, on the right side of the slide, you see the engineered authentic cell. This is something that's, as part of platform, is near and dear to my heart. And the hope here is that we can augment the cells genetically to be able to extend the platform, essentially, to be able to engineer those cells to do things like deliver payload and treat both rare and common diseases. And I should point out that we don't think all programs necessarily need that engineered functionality, but for certain indications, we think that it's going to provide a competitive advantage. So this is how our platform is structured. We start with high quality, healthy donor cell material and undergo reprogramming to lead to iPS cells. These cells can then be optionally engineered to make them universal cells, as an example, or to add payload to cells. But of course, it's an infinite number of things that we might do to make a more efficacious medicine. But whether it's authentic or engineered, they both then go through an expansion and banking phase. And once we get to that bank, we then induce them to differentiate in the top example to an authentic cell like a dopaminergic neuron, or on the bottom, like an engineered authentic cell like microglia expressing an enzyme. And I know in this crowd, you guys wouldn't be shocked to learn that it's not as simple as having the arrow go up and point to one cell type or another. But really takes considerable biological expertise in order to go from a pluripotent stem cell to a cell type of interest. And here, I think we're pretty lucky at BlueRock to have a number of different collaborators that help us on that journey. As Ross pointed out to us, one on the top here is near and dear to my heart. That's Llorente, who I've now worked with for over 20 years. Llorente is a co-founder of BlueRock and really has helped to fuel our neurology pipeline. Below Llorente is Bruce Blazar in the University of Minnesota, who's an expert in immune cell therapies. And below Bruce, you see Gordon Keller, who is at University of Toronto and has been really important in helping us push forward our cardiology program with ventricular cardiomyocytes. And on the bottom is the last row. This is a newer collaboration for us. It's a strategic alliance that we have with Opsis Therapeutics and their parent company, Fujifilm Cellular Dynamics. And so we're trying to push products forward in the ophthalmology space here. And so, I think, again, this is one of the key differentiators for our company, and I'm really proud of this, but trying to get that academic science and get it to a commercial phase is a huge task. And so at BlueRock, we've spent quite a bit of time now really focusing on building a world-class tech ops organization that has fully integrated CMC capabilities. And I'm going to not sit here and just read all the bullet points. That'd be pretty boring for you to listen to. But I do want to talk a little bit about the pictures on the right. We've opened a pilot cGMP cell manufacturing facility a few years back in Toronto. And right now there's a pilot cGMP gene materials facility being constructed in Cambridge, Massachusetts, that will be opening next year. And then on the bottom is, in my opinion anyway, the most exciting facility that's opening right now. This is the BlueRock Bear Commercial Scale Cell Culture Technology Center that's currently being constructed on the campus of Bayer in Berkeley, California. And many of you probably know that BlueRock is a wholly owned but independently operated subsidiary of Bayer. The independently operated part, really important to us. We are responsible and accountable for our operations, our pipeline, and getting them into the clinic and moving them through. But as we move towards commercial, I think there's a huge opportunity for us here to leverage the global clinical, regulatory, and commercial networks that Bayer has in place. And so for me, this center really represents the possibilities for both of our organizations in having the best of both worlds, really, in allowing us to be agile and move forward quickly, but really leaning on the muscles that Bayer has built. So, okay, so having said that, I want to drill down now, talk a little bit more about the reprogramming phase here. When you look at building a PSC platform, there's really these three different things that you have to consider. There's donor selection, there's the reprogramming technology that you're going to use, and then the cell banking platform. So I'm going to just run through these quickly. With donor selection at BlueRock, we're lucky to partner with Be the Match to identify our donors. And of course, Be the Match operates the most diverse and largest marrow registry in the world. But no matter who you're going to work with, if you're going to go make your own iPS cells, you're going to have to sit down and look through the inclusion-exclusion criteria. And actually, circling back to what Ross said earlier, I think for any other vendor you're going to go look at cells from, you need to know all of this stuff that is here on the slide. The inclusion-exclusion criteria, you might consider doing any number of things really to shape your funnel. There are different genotypes out in the world that, if pulled through into your medicine, could provide an advantage. So it's really tempting to try to shape the donors coming forward. You want to be careful to be not too exclusive when you do this, however, because any donor that does come forward still has to go through and pass an infectious disease testing and medical history review. And the donor, of course, has to consent for commercial use of their cells. Now, for us with Be the Match, they also collect samples in compliance with global donor eligibility requirements, which is great. And that material also must work with your reprogramming technology. And ever since the work of Takahashi and Yamanaka, of course, there's been an avalanche of different reprogramming technologies that have come forward, which is great. And here, I don't think there's any clear answers. But some of the things you should look for is selecting the tech based on the performance of that reprogramming platform. And of course, there's CMC considerations here. You want a defined process, if possible, that doesn't leave a genetic footprint. And I know there's a number of academics that'll be on this call, and that this is really the focus in some ways of this webinar. And this may be somewhat of a foreign concept, but I think you want to have some understanding of license availability for your field of use. If you're going out and you want to develop a medicine that's going to go to commercial, there should be a line of sight to how you could get a license for that reprogramming tech. You don't want to be locked out. Okay, so now we've talked about pulling donors forward and getting material to reprogram, and then you go through the reprogramming in a GMP facility. And as clones come up, as lines come up, some are going to fail on the basis of growth and morphology or other QC tests. But ultimately, hopefully, you get a number of lines coming forward that you're able to test. And so at this point, what I wanted to do was pivot back to what I think the intent of this webinar is, and reflect myself a bit. And what if I was to start from scratch in academia, again, trying to make a new therapeutic product? And I think the first thing that I would do is really try to identify as many clinical grade iPS cell lines as possible to be able to test. Now, keep in mind, even if you're not reprogramming yourself, you have to go through all those previous considerations that I put up here in identifying those cell lines. And then the next thing is really looking at differentiation potential. And when Ross flashed up the CIRM grant information, I was actually really excited to see assay development. I personally think that assessing the differentiation potential really requires you to have your analytical house in order and having a pretty granular look at what cells you're making. And doing that up front, I think, is going to save you heartbreak later on down the line. So in this thought experiment, I'm imagining that only some of the lines are going to have the potential to go forward, or you're going to prioritize moving some of them forward. And the next thing you'd want to do is what we would call end of production testing, or some variation thereof. And the concept here is that you'd want to make sure that those lines are going to be stable for many, many, many population doublings. And you'd want to model how far out you think you're going to need to go to make multiple batches of your medicine all the way out to commercial, if you can. And of course, what you're hoping to see is that your line is genetically stable well beyond where you think you're going to need it to be as you take it forward and grow your master cell bank and working cell bank and all the biomass that you're going to need to make different lots of drug. But in addition to being genetically stable, that they don't lose that differentiation potential as well. All right, with that, I will wrap up. Just say thank you for your time and attention. Leave you with my email address in case there are any questions after the webinar. Point out that hopefully if you're going to ISSCR next week, you'll come by and say hello to me. And then I'm going to shamelessly plug us and point out that we're hiring. We're always hiring. So check the website for more information. And thank you again.
Thanks, Mark. I'm glad you brought up the topic of the understanding what your ability to move forward with the line is. I've attended several conferences recently where there's been a lot of talk about that being an actual roadblock. People need to understand what they're getting into when they choose one of these lines and make sure they understand what potential they have for developing into a therapeutic. I'm going to introduce now our next speaker, Dr. Stephen Katman. He's an executive director at Sano Biotechnology in Seattle, Washington, where he leads multiple pluripotent stem cell derived therapy programs. Steve has extensive experience in pluripotent stem cell research and product development. This experience includes postdoctoral work in New York City and Toronto. We're using PSC, Steve identified cardiac progenitor cells, as well as the molecular underpinnings of cardiac development. Steve then went on to develop and manufacture products for life sciences and cell therapies from pluripotent stem cells at Cellular Dynamics International Fujifilm in Madison, Wisconsin. Steve?
Thank you so much, Ross, and thanks to our colleagues at CIRM and the other panelists to discuss this important topic. This is, as Mark and Jeff can attest to, this is something that we think about frequently, and it's really important to identify these lines and how your approach to looking at them early on in your product development, or for researchers while they're developing something that they want to take forward to the clinic. So just a little bit about Sano Biotechnology. So I'm located in Seattle, Washington, but we do have 3 sites, one in San Francisco and one in Cambridge, as well as Seattle here. And our goal is to develop novel therapeutics to repair and control genes in any cell in the body, but we're also developing therapies to replace any cell in the body as well. And these are obviously frequently from pluripotent stem cell backgrounds on these. And importantly with this, and touching on what both Jeff and Mark touched on for manufacturability, we're developing scalable manufacturing systems to enable broader patient access for these novel therapeutics as we go forward. So I'm going to kind of try to jump right in my intent here was to kind of jump right into the meat of what I've found from my experiences, particular pain points, both from postdoc work as well as working in other companies and even in here at Sano. So hopefully it'll be a theme going forward. But one way I would think about your PSC lines, I saw some of the questions coming up, and this is regardless of their ES or IPS cell lines, this is a critical raw material going into your allogeneic cell therapy product, right? So your starting cell line material is critically important, right? And so the quality perspectives or attributes of these cells are going to be carried all the way from your PSC to, as Mark talked about, with your master cell bank drug product, and ultimately into your patient. And so these are going to have huge impacts on your potential safety as well as efficacy. So I'm going to mostly touch on these as well as what we call genomic heterogeneity and differentiation potential. I'm going to steer clear of regulatory compliance and licensing. So there's a lot of information out there on donor eligibility criteria, which is obviously critically important, as well as licensing of the line. But there's multiple sources one can look for that. But anyway, so when thinking about using different pluripotent stem cell lines to develop your product, each of these are going to display unique safety and efficacy properties that result from this genomic heterogeneity and differentiation potential. And so again, these decisions should really be made early in product development. I know that's easy to say, really hard to do, because you're going to find a line, you're going to start working with it, you're going to be satisfied how well they differentiate in efficacy in your model. But as you progress down the product development pipeline, it just becomes more and more difficult to turn back and find that new line. So the two concepts that I'm bringing up here, are really no mystery, but these ideas of genomic heterogeneity, and this can come in many various forms, but such as single nucleotide variants that pop up, or karyotype abnormalities, anything like this. But these can be either donor-derived. So in the case of iPS cell reprogramming, of course, these could be mosaic in the starting cell population of your somatic cells. And during reprogramming, you might find clonal variants of these genes, right? So one clone might harbor it and another clone might not. If it's from an ES cell line, obviously it's most likely going to be more uniform across the whole population. You might find various clonal variants in there, but these could also be culture derived too, obviously. So Mark touched on, you want to make sure that your PSC line can maintain a stable genomic profile throughout many passages as far as you see the product development going, really, and maintain its differentiation potential in there. So, early on at your starting cell line population, you might find no concerning variant detected. These might arise possibly through your culture conditions, and possibly there's potential there that they might be variable. There might be some sort of propensity there, or likelihood that they're going to become a variant. These things are all to be discovered. There's many researchers working on this right now. Oops, apologies. Back a couple slides. So I'm just clicking around my tiny screen on this. And so obviously, you have to care, one should look at these variants and use a weight of evidence approach to figure out how they might impact their product, including the safety and efficacy profiles of their product. But, some of these may even expand in culture or even in vivo post-translation. So you really need to think about these. So importantly, what I want to point out is this heterogeneity occurs naturally in humans and in cell culture, and it could be passed from the donor or emerge during cell division. And so this gets to Mark's point as well, like really strong characterization of both your PSC population as well as your differentiated product. And I completely agree on the emphasis on assay development. It's one of my favorite things. I love assay development for these because we learn so many new things from the cells. So this heterogeneity, as I touched on earlier, this may affect affect your oncogenic potential. So for example, having uncontrolled cell growth, this would obviously impact your product and your path to the clinic. There could also be differentiation blocks, and this might be partially through the differentiation process or not being able to get there at all. And so thinking about these early in the development and what pathways are being hit by any potential variants is really important. This should be, from my experience, one should characterize these and really understand them well. And each line obviously might have unique genomic heterogeneity profiles, potentially affecting this oncogenic risk and differentiated cell phenotype, right? And so each researcher organization needs to think about this for their development or differentiation process, as well as their approach to the clinic, and use that weight of evidence approach to look at it. All right, let me advance the slide. All right, so the next thing that could be impacted, which might be unique from line to line, and this includes both PSCs or iPSCs rather, and ESCs, is the differentiation potential. So while redacted, I'm still, going to use this example of two different PSC lines. These are actually closely related PSC lines, and it's important to build robust differentiation processes but I think Jeff pointed out too that, you know, little tweaks in the process are going to be necessary for each line. So I've seen a lot of publications out there where you look at a, you know, narrow window of a differentiation process, and one says this line can do it and this line cannot. But that's inconsistent with my experience, in which, you know, I agree with Jeff, where you can take a line, and as long as you understand the developmental milestones and have the assays for those different points during the development, you can modulate the manufacturing system to create that line. And so these are going to display different. So here's an example of different potential between two different lines using very closely related manufacturing processes. So if we look at just inductive factors, so like kind of your key molecules or key components that you're adding into your manufacturing or differentiation system, these might change from line to line. So in this case, this line requires a lower concentration of factor X. And also the target population the percentage of cells arriving in the target population might change. And so even despite our best efforts with this PSC line 2, we might require a selection step in there to get to our final purity of cells that we want. And changing your manufacturing could change the comparability of your cells at the end. So it may have no impact at all if you change some things in your manufacturing process, but you should be, you know, aware of that and make sure you have a suite of potency assays and characterization assays that you're looking for to be sure of that. And next slide. And here's another example of what could change besides just the differentiation potential. So this is looking at another two PSC lines, so line 3 and 4, as well as, you know, these are the differentiated lines from these particular pluripotent stem cell lines, looking at the target population in this salmon color, I guess, and the off-target population. So these are the cells that you're after, what you anticipate are going to have the efficacy. You can see this is using very closely related manufacturing systems here. You can see that the target population purity was very high for each of these populations, and off-target as well, so being below 5% in this case. But by using single-cell RNA sequencing, then in characterizing that product, we can see that, you know, this is just this cluster analysis here is only based on that less than 5% of cells, so not the target population, but only the off-target. What you can see here by the cluster analysis is that there's populations represented from both lines 3 and 4, so very similarities in there. But interestingly, only line 4 had a unique population in it. And what impact might this have on your particular therapeutic that you're developing? This is where this characterization becomes really important for what you're doing. And importantly, like if you change lines later in your development process, you'll have to understand that and be able to understand that potential risk that comes up and mitigate for it too, and be able to characterize it. So the single-cell RNA sequencing can reveal these unique populations of differentiated products. And this could impact your comparability, safety, efficacy between different PSC lines. Move forward. So just in summary then, different PSC lines may display unique safety and efficacy profiles as a result of this genomic heterogeneity and differentiation potential. And, you know, I also want to emphasize here that we really have, you know, the impact on this is obviously both safety and efficacy, but if you change lines later, it can change how you're looking at the comparability of those lines and how much characterization you need to do with them. And so these cell line decisions should be made early in product development. How early, you know, it's hard to say for sure, but product development in order to maximize understanding of safety and efficacy profile, as well as minimize comparability risk between the lines. Let me see if I have one more slide. Oh, just thank you. So thanks again to CIRM and our fellow colleagues on the panel. Looking forward to the discussion.
Thanks, Steve. I appreciate the presentations. We're now going to move to the discussion portion of the webinar, and I'm going to just introduce Dr. Irene Llorente. and she'll be moderating the discussion. We do see that there's a number of questions that came up in the question and answer section. I think we've answered quite a few of them live, but I'll try to get to the ones that I think we didn't address up to now. Dr. Llorente did her predoctoral training between the University of León in Spain and the University of Florence in Italy with an international fellowship from the Spanish Science and Innovation Department. She's now a research assistant professor in the neurology department at UCLA and a faculty member of the UCLA Broad Stem Cells Research Center. The main focus of her research in the past 15 years has been largely directed toward understanding the biology of white matter repair in the central nervous system disorders, with a special emphasis in human glial biology. Dr. Llorente has developed unique allogeneic human-induced pluripotent stem cell-derived glial-enriched progenitors cell therapy product for the treatment of white matter stroke and vascular dementia, and is currently funded by a CIRM grant to finalize product formulation and in vivo confirmatory testing to enable a pre-NDA meeting with FDA. So thanks to the panelists for hanging out with us here. Irene, do you want to go ahead and lead off with a question for the group?
Absolutely. First, I want to thank our three speakers because the talks were fantastic. And I want to thank also CIRM for creating this webinar that I think is highly informative to all the faculty and also the biotech companies that we are moving towards a clinical application with cell-based therapies, right? And my first question, I know it's a really difficult answer for the three speakers, is you guys have been talking about variability between different cell lines, tweaking the cell differentiation protocols, etc. My question is, how many cell lines should we screen to create a final therapeutic product? Are we talking about 10? Are we talking about like hundreds of them? We're all limited by financing, basically. And also, when do you test all of these cell lines? During the differentiation stages, during the efficacy testing, during the manufacturing? Please, guys, enlighten us.
I guess maybe I'll jump in here and start us off. I actually, as part of the slide that I left in the end, I was going to make an excuse for it and point out that I know it was really ugly, that it had those arrows all over the place. And that was actually a stylistic decision that I purposely left there because it represents a lot of work and a lot of time. And part of what I was going to point out is whether you're in industry or in academia, it's a big work package to commit to. It's a huge amount of work. And your question is exactly kind of the point of that slide and the gradient I left in there, which is to say, you kind of want to test as many as you possibly can. And you might even be lucky enough to find yourself in a situation where the line you've selected actually works better than whatever reference you were using in the past. But on the flip side of that, again, I think to do that experiment well is a huge amount of work. And so it's a tough decision to make early on, but I think it will be a wise decision down the road. It will save you from heartbreak downstream, where things are more dire and you've lost time.
I completely agree with that. I think as many lines as you can get your hands on, and it makes financial sense for you, and as soon as possible, because you really want to lock in what line you're going to be working on so that you have as much time between experience with that in order for you to be able to put ttogether the best IND package that you can. And especially if you're going to need to re-optimize some here, you want the most amount of time to be able to do that, but the most amount of time to know if there's going to be any safety issues or off-target populations or things like that that might be cell line specific. So I would say as soon as possible, as many as possible, are feasible.
I would say I can completely concur. However, the obvious challenge there with as many as you can is you're going to be limited by something, right? Like you're going to have the hands in the lab to do it, or the funding to do it, or both, right? Like, and so that's always going to be a challenge. And so and especially if, as Jeff was talking about, like if there's particular lines that you might be able to tweak the differentiation of the cells, right? Like, and this is from our experience too. It's not like an inherent ability to make islet cells or cardiomyocytes or neurons or anything like this. You tweak the methodology to get there. And so like doing that for, 100 different lines is obviously going to be a huge burden. So, you might want to figure out a stepwise way to do it and screen it out. Speaking to one of Mark's slides that we saw there, screen outlines early on, right? Like what are going to be no-goes, right? Is there going to be a genetic variant that's absolutely not going to be acceptable for you? Then you can start to get into differentiation, make mesoderm, make endoderm, ectoderm, et cetera. So it's tough, but the important part is early, do that screening and make those decisions as early as possible. It just gets harder to do later, right? Backtrack. It's not impossible, but it's hard.
Thanks, guys. I appreciate the comments. That's a question we try to consider at CIRM. We try to put a number around what we can provide as additional support. We recognize that's an additional cost, and that's why we made the additional supplement available. We tried to come up with a number that seemed reasonable to us. I guess time will tell whether we provided enough funding to actually ask this question appropriately. We hope we'll get better outcomes from it. But all those considerations are things that we are concerned about. And that's why we're focusing on this stage to try to get our investigators to be able to access additional lines during this stage. I'm going to ask a question from the audience right now. There's one from Mark Schuyler Scott, who wrote, if protocols need tweaking dependent on cell lines, how would this be viewed by FDA? Is the process control as important as deeply characterizing the final product? Anyone wants to try answering that?
Yeah,
that's one of my favorite questions. Oh, go ahead, Jeff.
No, you do it, Steve. I think we're probably going to say the same thing.
Right. I think this gets to Mark's point actually about making sure really good analytics, right? So if we've heard often that the product is the process. Comments like this. That may be the case, but the product's the product at the end of the day, right? So you should have a really well characterized product at the end, and there might be a need to tweak the process within it. And so this is where like your final product or drug product characterization is really, really important. And so it's hard to know how. It's going to be a case-by-case basis, from my understanding, that the regulators are going to view it. So if you can tweak the process a little bit and it doesn't, you have ways to show that it doesn't impact your safety and efficacy, I feel good about that, but I can't speak for the regulators on it. Okay.
And if you're going to go forward with an allogeneic process or a product, you're probably just going to have one cell line. And so that's why you want to choose one cell line, optimize and do all the work and characterization that you can with that one cell line. Maybe also a implicit thing from the three talks. I don't know that anybody actually said explicitly. I think it might be a harder situation if you're talking about multiple cell lines, or maybe like an autologous iPS product. That becomes kind of a nightmare. I don't know, because you would basically, I don't know how you would approach the FDA saying that you're doing an autologous iPS line and you need to do individual tweaks for each individual iPS line, unless you came up with some sort of like analytic or universal protocol or something like that to allow for you to not need to actually do tweaks, or to be able to do tweaks in a very direct way, as opposed to like what we do currently is more shots in the dark and kind of seeing what ends up working there. So just wanted to acknowledge that.
I'll just quickly agree with both of my colleagues here. And the one thing I would build into that is something we also said, which is pick that cell line early, and then you don't have to bridge across. You never have to have that conversation with the regulators to begin with. This would only really be a concern in an allo setting if you did a whole bunch of work and picked later, and you wanted to reach back through to the other data, right? So as long as you made the decision early, you shouldn't even have to make that known.
Thanks, guys. Irene, I'm going to ask one more question from the audience, then I'll turn it back over to you again. So the next question is from Jonathan Rodriguez. In your pipeline, and I assume this was to your talk, Mark, initially, but I'd like you all to consider it. In your pipeline, you're first mentioning high quality, healthy donor cell material. I'm curious how you discriminate and screen high quality donors. And I guess they're asking about the cell lines themselves. What are the defined criteria that you use?
Yeah, to the last question, I'm not going to tell you exactly what we do at BlueRock, but I can just broadly say, I mean, there's the medical history, right? And then so you could consider whatever you'd like to consider as far as medical history, family trees, how has the family been over time, things like smoking or body mass index or things like that could all be considered as part of what you'd like to see and bring forward. It's really up to you to define it however you want. But in the case where you're sourcing those cells from someone else, it's probably wise to try to see how they defined it.
Steve or Jeff, did you have any other comments you wanted to make about whether you consider the donor material itself or?
I thought Mark did a great job.
Okay, great. All right,
Yeah,
Irene, do you want to go ahead with another question?
Yes, I pick a question from the Q&A in the chat because you guys have talked a lot about analytical markers, analytical like testing. So Kevin Demore is asking, do you have or are you pursuing analytical markers, DNA, epigenetics, gene expression that predict performance among PSC lines? Or is it just most reliable and pragmatic to test differentiation performance? For any of the speakers.
I'll take a first crack at it. So the way we do it currently is we simply have to empirically look at differentiation performance, which is quite awful because our differentiation takes a month and it's quite costly as most differentiation procedures are. We do have some kind of in-house know-how in terms of like testing out usual tweaks to the process that we usually use as kind of a screening tool to give us a better idea on it. But a lot of it really comes down to kind of a cell whisperer sort of approach. So like you have the very talented operator there that's like, oh, the cells look this way at stage 3, and therefore I got to do, 2 years ago, I did something in response to that, and that worked out for a different cell line. With that said, we're currently working with a team to try to actually get some characterization work to be able to either be able to take a new cell line, get a measurement, and know is this going to work well or not with like with a given process, or alternatively be able to look at a cell line and say out of maybe like 3 options in terms of protocol tweaks, this would work best with option 2, for example. We are currently in the process of doing that with a collaborator with a bunch of various omics approaches for doing that. Though I'll say that we're still, that project is still very far away from producing anything that is tangibly useful at this point. It's hard.
I'll take a quick swing, and hopefully people don't hate me for this. I'll tease and say, I think we've got some analytics that will tell us. It's early. I reserve the right to be wrong later, but so far it's looking pretty sweet. And I would love to be able to tell all of you about it, but of course I'd be in big trouble with legal. So I'm not going to be able to do that. So apologies, but I think it's doable.
I think, yeah, and to follow up on Jeff and Mark too, I think Kevin that asked the question, you would know this very well, of course, too. But like I think there are predictive tools out there, and those have some utility, right? Like for the scorecards and things like this. But I think for any organization or researcher, knowing the major levers in your process, right? And then is there anything about the pluripotent stem cells at the PSC stage that impact those levers, like those drivers of differentiation, BMP, Wnt, activin noodle? It's, but at the end of the day, it comes down to, at this point in time, at least, it's empirical. Like you just have to test it.
Irene, go ahead and pick another one.
Yes, I would like to ask you, this is from Drew Sarin from CIDARS, and he is asking, can you shed some light on the variability of the raw materials, cytokines, small molecules, etc., from different vendors, and even different lots from the same vendor? How do you qualify these critical raw materials for your particular differentiation process, activity, purity, etc.? These are some of the biggest contributors to the variability. How CIRM can address or help addressing this challenge?
Start by minimizing. Don't use anything you don't have to, number one. And then number two, you know, like if it's, you know, if it's a critical raw material that could influence the differentiation potential of your cells, you know, you probably have to develop an assay for it as you're taking it in and screen for it. And that might, you know, you might need to develop something where you change the concentration of it or things like this. And that's sterility and other things that are implicit aside, right? Like, but
Yeah, totally agree with that. I would say, you know, for critical raw material, you'd love to have that assay built. Be checking each lot as it shows up. Check the stability in the freezer. Check the stability out of the freezer, et cetera, right? You'd really want to look at every critical raw material and how you use it. Anytime you're switching vendors, of course, right? I mean, that gets very important. And I guess in my experience also with GMP-based reagents, for reasons that don't always make sense to me, they're usually not as good, which then creates another obstacle for you to go forward on. But I think if you have an assay and you could bridge across and understand what the actual activity is that's in the tube, as opposed to just reading what's on the C of A, you'll get much further ahead and you'll be able to figure out those bridges more easily.
And just to add to this, again, I'm coming from an academic perspective here, so we maybe don't have like certain quality issues that industry has to worry about, but maybe we should be worrying about that more in academia. One of the things that we've done is, as much as we can, switch over from using growth factor cytokine-based reagents to small molecules when it has a possibility. Just practically, we find that vendor variation or batch variation of small molecules is tiny versus what we've experienced with growth factors, and they also tend to be cheaper, which is kind of nice when you're running a academic lab, though I guess we all have cost issues to worry about there. So that's one, and we've done that as much as we can, though there are two things that we still haven't switched out yet, which is very unfortunate. The other thing as well, which is maybe kind of a little beyond the question, is how you actually handle the reagents in the lab matters a whole lot. I can tell you that like my team at WashU, we've really implemented very strict handling instructions for everything, and it seems to matter like a whole lot. Like only using single-use aliquots and keeping everything in the -80. No, don't freeze thaw or thaw freeze or anything like that. And that's at least in our experience does greatly help to increase the efficiency of our differentiation process.
And I'll speak up for a second here also, since the question was, how can CIRM help with addressing this challenge? And the answer is, we're a funding agency. We unfortunately don't have our own labs to test these things out. We have plenty of investigators that are asking these questions, but unfortunately it's up to the investigators themselves to share this knowledge amongst themselves. You know, we can try by organizing workshops like this to bring together discussions of common questions and try to talk about ways to address it. And that's what I think we can do in the short term, but we're certainly aware that this is a challenge and it's something we would like also dealt with. So we appreciate the contributions from the speakers today answering these questions. I'm going to go ahead and pull a question out of the queue, actually. There's one from, I guess the name, I see initials, ZD Shi. The question was, can you engineer research grade cells first and then adapt them to GMP grade afterwards to generate master cell banks? Or do you have to start with clinical grade in the beginning? And I think I know the answer, but I'd love to hear your short answers to this. If anyone wants to try.
it depends. That'll be, you know, what you'll hear often is it depends. It's, you know, it's not, it doesn't, from my understanding at least, it doesn't have to be GMP from the derivation, right? And there's plenty of examples of this in clinical trials right now.
My guess is it's what you bring to the FDA, and the FDA will tell you what they expect to know about it if you want to bring it forward generally. And if you start with the GMP run, the questions you get asked are less. I assume that's the reason that you want to try to start this in the beginning. The fewer questions FDA has about the product, probably the better for your path forward. Irene, do you want to go ahead and get another one?
Yes, this is a follow-up from Kevin Demore, and it's a question that I also asked myself, and I'm changing gears a little bit about PSC different clones. So basically, for the people that they are considering gene editing PSCs-based products, how you take into account and the different clones, do you test them in efficacy? How many of them you tested? It's kind of like similar question in line with how many cell lines. Also, how many of those different clones are you also testing in your manufacturing?
I think the answer is maybe going to be similar to the cell line selection, that you want to test out multiple there. And clonal selection, it has its own challenges though, because by doing by clonally growing cells out, you could potentially be, and theoretically can be increasing the likelihood that you're selecting clones based on mutations that give an advantage for them growing out as a clone. So that's maybe one difference there. But in general, my experience, clone variation tends to be, in terms of differentiation performance, tends to be less than cell line variation. So maybe there's like some things that are you're less worried about, some things you're maybe more worried about. But I think you kind of, in my opinion, you kind of end up in the same place, that you want to test out as many clones as is feasible, both for your differentiation process and function of the drug product, along with safety and other sorts of concerns.
Yeah, I just quickly agree with that.
As opposed to testing everything in your suite of assays that you might be using for your primary line selection, though, you should probably be able to narrow it down a little bit, which might be more unique to the subclones, but it's case by case basis, I would say.
That's a really good point. If you went through this experience with the cell line selection, things that you maybe didn't appreciate as being problematic, maybe now when you're actually going on to the clone selection, you can appreciate that more and have a better analytic strategy based on that experience. Hence, like what we said at the beginning about like doing this as soon as possible kind of helps to build on itself.
Thanks, guys. I'm going to take a question from Lila Collins. Her question is, it seems that the key tweaks required between lines is at the germline stage. Is it possible to force all pluripotent stem cell lines to some sort of shared baseline pluripotent state and harmonize from there?
I'll take a crack at that one. Theoretically, yes, but practically, I don't know really how to do that. I mean, there's a lot of literature out there at this point on different states of pluripotency, and we have a lot of different like just commercial reagents for growing these cells that likely lead to there being some difference in how stem cells are, the state of the stem cell. So I think theoretically, yes. Practically, though, I don't know how to translate that theory into practice at this point.
I feel boring for just agreeing, but yeah, totally agree.
It would be wonderful, but I think people have also developed, like if you have different, say, reprogramming states of cells, and methodologies have been derived from different users on those different, reprogramming states, or what might be conferring that difference between lines. It's going to be hard to find something that's uniform with it. United Nations of PSCs.
Irene, do you want to pick another one?
Yes, Jonathan Rodriguez is asking, one of the speakers has mentioned automation during one of the talks. With these tweaks that are required between cell lines, how an automated system will handle, in your opinion? And in other words, how reliable will an automated system be with this highly variable donor wall in manufacturing reproducibility point of view?
Yeah, and I'll preface that by saying, and you maybe kind of picked that up on that based on my tone of voice, that slide was out of order. So it kind of like came in the middle of it. I don't know how that happened, but I did the best I could to adjust to the slide being out of order. So it gave kind of a false impression. So, so automation, at least the way that we're pursuing it right now, would really help to reduce variation in the context of batch-to-batch variation and user-derived variation. What we're pursuing here isn't really designed to, basically you would need to have, using like a manual process, have figured out what is the tweak necessary to make whatever given cell line you have differentiate well, and then just have the robots go and do that instead. So again, my order of slides made that very confusing. I realized it's really for batch-to-batch variation, not cell line variation. So with that said, though, I mean, I do think there's a place for automation, maybe in combination with like machine learning or something for that approach to be used to be the actual troubleshooting, if you want to really brute force this to get, you know, brand new cell lines to work. So there is a possibility for that with automation, you know, again, perhaps combined with machine learning, though that's not something that we're pursuing at this time.
I guess just to build on to that, Jeff, you could imagine, I think, a situation in scale down where maybe you had automation explore your favorite tweaks of interest that you had up on your cell lines, right? So that could be a road to go down. But yeah, I think for this crowd, you know, that's a heavy lift.
And grad students are a lot cheaper just to multi-wells.
Yeah.
Oh, go ahead.
Do you want to pick, or should I pick another one?
I was going to just take Drew Serene asked a follow-up question. What are the panel's thoughts regarding picking a single clonal iPSC line to make a GMP master bank versus a polyclonal iPSC line bank to make a GMP? So would you choose one or the other, or is there a reason to do that?
I think clonal simplifies characterization, right? And so, especially if you have any mosaicism in there, you know, I would prefer to go clonal, but there's going to be potential caveats to that too, what you're doing, right?
Agreed. And I would just add on that many people feel very strongly about, and none of us should speak for the agency, but many people feel like the FDA really wants to see clonality in banks. And I think in many ways, this comes from larger biologics and CHO cell banks and probably the history back there. But there is a thought that some have that clonality issue is big. So I don't know how they would react to a polyclonal bank, but you know, even without having to resort to speaking to the agency, I think Steve's point is spot on from where I stand as well. It would be much easier.
Irene, did you want to go ahead?
Yes, Tom Allison is asking, is there a centralized effort to look at transcriptomics or other cell line parameters to be able to predict what makes lines good at, say, beta cell differentiation to allow for a more direct approach?
I don't know there's a centralized effort, but I remember referenced to this earlier that we do have an effort right now on using various multi-omics approaches across cell lines to see if we can figure out these things. So it's something that at least we're doing. I wouldn't be surprised if other teams are trying to do it. I think Mark made a mention to having a secret thing as well for that. So I don't think it's centralized, but I think people do care about this and are looking into it.
I'm realizing now I'm never going to live this down. But you know, the one thing I would say about this too, that's maybe a slightly different angle, which is I'm not sure that any line, and this was sort of alluded to by somebody, maybe with Steve, I don't think that there's any line that necessarily, well, there probably are some, I shouldn't make a global statement like that, but it's probably not the line, it's the line with the process, right? So it's the two together. Most of these processes have been developed around a given line. And so that's what it is. And the point has been made that you can fix the process, right? You can make it adjust to the line. So I'm not sure that the approach would even work other than unless you're really anchoring it to one approach, one bioprocess that everybody's using, because otherwise it might contaminate the data that you're trying to collect.
Yeah, that's where I was going to add to Mark's comment there. Like, this is exactly where I think it's more beholden on the single user, right? Whatever company or academic to do that specific for their own needs, as opposed to like a concerted effort going forward.
I'm going to take this next question from Marcos Valderas. Probably mangled that. I'm sorry for that. And this is not an easy question to answer. So I guess I just want to ask, what do you think about for considerations of doing this? He's talking about how you choose how big of a master cell bond to make. Considering diseases by case scenario in terms of scale, what is the ballpark estimation for the number of patients you should reasonably anticipate treating? And so I think the question is more about when you're thinking about making a banking process, you get your master cell bank, you make your working cell banks. What's the scale you should be thinking of, and how do you try to determine that? I assume some of it's looking at the patient population, but what are the sort of exercises you imagine doing when trying to address these questions?
I can take.
I'd say work backward. Oh, sorry. Go ahead, Mark.
Oh, go ahead, Steve. You're going to actually give the answer that I think I'm going to give, so go for it.
Oh, yeah. I was just going to say work backwards. You know how big your indication is, how large is your dose, how what's your yield of your drug product that you're making. Then you've got then you can work back to your PSC banks and then talk to somebody in tech ops, because they're going to up that number a lot, because there's going to be many that are needed for retains and analytical testing. So it's probably double what you ever think it'll be. And then, but that's important to think about too, for like bioconfiguration and all these concepts. So how big is it? How is it configured to? So how many units do you have? How many cells per unit? All these things weigh into it.
We're actually getting a little close to the end of our webinar now. Irene, I wanted to give you a chance to ask any other questions you might have thought of that you wanted to bring up. Was there a last question you want to ask the group?
I do. Somebody has asked what I think is a really good and fun question, because we have been talking about allogenic cell therapies all morning, right? Brianna Kalikaran is asking for advice for autologous products. And I feel like we have avoided the subject for a bit. So I wanted to give you the opportunity to talk a little bit for her and other people about autologous products and your advice on them.
I'd love to jump in on this one, because I've always, this isn't speaking for my current organization, because we're developing allogeneic for very important reasons, but I don't see, manufacturing wise, I don't see a reason that autologous can't be done, right? So I think there is possibly potential there, but then it comes down to regulatory questions as well. And I think those would probably be considered a little bit differently than allogeneic products, because it's the same cells going into the same patient, which is a different scenario, right? And so I think early discussions with the FDA would be important for you there to see what applies and what doesn't.
I'd add to that that basically everything that we have said here applies much more importantly to autologous. And the big disadvantage or the advantage that allogeneic products have, in this context at least, is that it's one cell line. You can do all the analytics on that one cell line. You have to process that cell line. You have a lot of time with that one cell line, and there's some forgiving there, because if something's wrong, you get a chance to go and fix it or change your process or whatever. When it comes to autologous, if you want to have this work on any sort of scale, you need to make sure that your processes and your analytics are spot on, so that you'd be able to go from the patient sample, make the iPS cells, make the drug product, do all the analytics along the way, and have that work most of the time on a single shot. So autologous is just a lot less forgiving, and you need to make everything work even better for it than for allogeneic.
Okay, on that, I'd like to thank our speakers and our moderator here for helping us through this discussion today. I really appreciate the lessons that you shared here. This is a difficult question to face, how to select an optimal line, but it's a key one. And we recognize that that's really an important step to figuring out what you're going to go forward to the clinic with. Switching later has huge associated costs. We all agree that you should really try to answer this question as early as possible. And I hope that what you've heard today in the audience has been helpful for you when you think about this process. So thanks again, everybody. It was a great session. And I should also say that we didn't get to all the questions in that were asked in the question and answer area. We'll try to get some written answers out as we can. But thanks for a great session. Okay, goodbye, everyone.
Thank you.
Thank you.